name is Lydia, and the project I'm working on is developing a CD microfluidic device for capturing and isolating cancer stem cells from non-tumorigenic cells. Alright, so here's our team. It's pretty small, but our team consists of myself as the team leader, Guillermo, who's our team researcher, and we are being advised by Dr. Kulinski and Dr. Ding. So before I go into talking about more details about my project and the overall design, I thought it'd be a good introduction to talk about cancer stem cells, since cancer stem cells are the main focus, and it's what our project is really centered around. So cancer stem cells are like regular cancer cells, in the fact that they are able to self-renew, differentiate, and grow rapidly. However, the main differentiation between cancer stem cells and cancer cells is their ability to form tumors. And that characteristic is what causes cancer to be persistent and in a patient's body. So, cancer stem cells can travel throughout different locations in the body. So a patient can develop tumors in, say, their lung, and the ability for this cancer stem cell to travel and differentiate into other cell types. So say, for example, if it starts in the lung and then it travels to like the liver, it can start to form tumors there. And that's the reason why cancer is still present in an individual's body. So as this diagram kind of shows, so there's a bunch of cells, and that yellow dot thing represents the cancer stem cell. So conventional cancer therapy, or what's traditionally being used in like chemotherapy and stuff, they don't target cancer stem cells. So as shown in the bottom half, when you don't target the cancer stem cells, the tumor still exists. So even though a patient will go through chemotherapy, after chemotherapy they might be like, oh, okay, now I'm cancer-free, but a few months later, a few years later, they'll develop cancer again. And the reason for that is this cancer stem cell presence. And the top half shows that a therapy or treatment that targets cancer stem cells will be able to diminish tumors. So it's kind of that step into solving or understanding cancer better. So another characteristic of cancer stem cells versus regular cancer cells is their difference in cell membrane capacitance. So cancer stem cells have a higher cell membrane capacitance than regular non-tumorigenic cells. And so we're going to use that property to our advantage in this project. Okay, so a bit of background about the project. As the title says, our goal is ultimately to create a CD fluidic device that will separate cancer stem cells from non-tumorigenic cells. Some of our objectives include separating about 90% of cancer stem cells from non-tumorigenic cells through a phenomenon called dielectrophoresis, or DEP. Another objective is to use temporal separation to separate the cancer stem cells versus regular cancer cells. So for our design, we are looking into using two different types of prototypes. So another of our objective is to determine from the two which prototype would provide the best results and prevent leakages. So why is this project important? What's the significance? Ultimately, this device will be used as a research tool. So researchers and scientists that are focused in cancer research, so some pharmaceutical companies that are developing cancer treatments, they could use this device to understand cancer stem cells more so that they can develop treatments and therapies around it to ultimately conquer cancer. So here's a simple SolidWorks model of the prototype. So it's the bigger glass on the bottom, that's a glass slide, and on top of the glass slide we're going to pattern gold electrodes, and then the other two layers, it varies between the two designs, which I'll explain. So as I mentioned before, we're using the phenomenon called DEP, or dielectrophoresis, and DEP creates a non-uniform electric field. As I previously mentioned, the cancer stem cells versus non-tumorigenic cells have a difference in cell membrane capacitance. So with a higher cell membrane capacitance, it's going to gravitate more towards the sides of the channels because of this electric field. And in order to create this DEP phenomenon, we designed two different types of electrodes, a rectangular shaped and trapezoidal shaped interdigited electrodes. And the reason why we did 2 is to determine from the 2 shapes which one would provide like the strongest field or to promote better separation. And so we're going to collect the cancer cells and the cancer stem cells through time, so a temporal separation. As the fluid and the cells move through the channels, it creates like a velocity profile because the cancer stem cells are going to move slower since they have a stronger affinity for the electric field on the sides. And then towards the middle, you have the non-tumorigenic cells that are going to move faster. So based on this time relationship, we can capture the non-tumorigenic cells first, and then after some time, we're going to release the function generator, like release the electric field, and then capture the cancer stem cells. So that's our method of capturing. So the 2 designs we are considering, it's based on material. So like I said, there's the glass slide, and then on top of the glass slide is gold electrodes. And one of our design ideas is to use PDMS, so make the channels out of PDMS. And after talking to some graduate students who have worked with PDMS and bonding it to glass, they've had some difficulties. So one of our ideas to combat this issue is to cover the glass slides and electrodes with a thin layer of PDMS, and then through plasma bonding, we can bond the PDMS channels on top of the PDMS, thin PDMS layer. So it's kind of the top 2 layers shown in this model. And the other idea is to use plastic and CNC machine the channels in it and to attach it to the electrodes and glass slide through a pressure-sensitive adhesive. So those are the 2 ideas we are going to test and see which one would provide for the best results and allow fluid to run through easily. Current work that we've done, so because DEP is a bit of a new thing for us, we wanted to observe it better before actually testing it so we know how it actually works. So we tested DEP on a sample device, and instead of using blood samples, we used polystyrene beads to mimic the behavior of the cells. So Dr. Kolinsky had a sample electrode device, and I brought a solution of suspended beads, and we just dropped a bit of solution on top of the beads, and we attached a function generator onto the contact pads, and it created this electric field. Under the microscope, you could see some separation between the cells, so that was pretty cool to see. Also, the graduate student that's in my lab had a sample device that we were also able to observe DEP through channels. So actually fluid running through channels and see some separation. Also, before testing plasma bonding on my actual devices, I wanted to see if it would really work. So I did 2 samples of plasma bonding. So for the first sample, when we took it out of the plasma bonding machine, we noticed that there were some air bubbles. So it was a bit obvious that fluid wouldn't run through, but I tried it anyways, and there was leakage. It just didn't flow through the channels. But I tried with another device, and this time we patterned it, we bonded it better. So fluid was able to run through the channels well, and I used green dyed water to observe how water flows, and no leakages, so that worked. So we can say that plasma bonding should be able to work when we do it with polystyrene beads or blood. Also, I designed the 2 types of electrodes. So there's the trapezoidal shape and the rectangular shape, and we are going to see which one of those would provide for a better electric field and separation. Future work. So now, actually yesterday, we just got the glass slide through the electrode. So that's pretty much ready. Hopefully tomorrow or next week, the plan is to start testing with the actual PDMS channels and the CNC machine plastic channels to see which ones would work the best. And we're not going to start right away with blood. We're going to first test with polystyrene beads, see how that reacts, and see how the fluid moves through the channels. And if that works with that and we're able to see a separation, then we can move on with the blood sample, which we're planning on doing for spring quarter. So budget, it doesn't really require too many materials, so we received $400 in Europe funding, and a lot of the materials that we are using are already supplied in our labs. So the only thing that we really had to spend money on so far is the mask, which costs about $65, and to use the clean room facilities to actually photolithography and pattern the design onto glass slides. So we might have to buy some more beads because I believe some of the beads we have are a bit old, but I think they bought some new ones. So just in case, we did encounter some other items that we would need. In terms of documentation and communication, to document our work, we have lab notebooks, so every time we go into the lab and do something related to this project, we record it. We also communicate mostly through email, and every Wednesdays we meet with our advisors for weekly meetings to talk about our updates, progress, and future steps, or what we're going to do next. And anytime we have questions, this is also time for us to bring it up with our advisors and see how we can approach it or solve it. So overall, I guess to conclude, how is this project innovative? There are already some devices out there that use DEP to capture and isolate cancer stem cells. However, what's innovative about this one is that it uses a CD, so that it promotes cell separation based on their densities in the beginning. So that promotes a little bit of a better separation between the cancer stem cells and the regular cancer cells, since they have a difference in their densities. Also, we want to be able to run more volume of samples through in order to collect more cancer stem cells. Some of the devices out there, they use very small amounts of volume to run through the channels. And because we're using a temporal separation, hopefully we can be able to run through some more fluid. Also, we want to achieve a higher efficiency or separation between cancer stem cells and non-tumorigenic cells. So that's ultimately why it's important. Some things we learned, pretty much since I'm not really bio-related, a lot of this was pretty new for me in regards to learning about cancer stem cells and learning the difference between cancer stem cells and regular cancer cells or non-tumorigenic cells. Also, for me and Gamma, who's my teammate, the phenomenon of dielectrophoresis was really new, and understanding its applications and how it can be used in cell separation was pretty interesting for us to learn, and it was pretty cool. Also, understanding the different methods of bonding materials together. Since we're testing two different prototypes with PDMS-based and CNC machine-based, it gave us a better understanding of what kind of materials are out there and how we can fabricate and produce different types of prototypes, but with the same goal, essentially. And that is my presentation.